Hello everyone, welcome to the course Agricultural Microbiology. Course number is PLP 101. Credit hours will be 2, 1 plus 1. That means 1 theory and 1 practical class in a week. So let us begin. At the outset, let us see the syllabus of the course. Here we can see the theory part of the syllabus, introduction, microbial world, prokaryotic and eukaryotic microbes, bacteria, cell structure, chemoautotrophy, photoautotrophy, growth, classification and reproduction, bacterial genetics, genetic recombination, transformation, conjugation and transduction, plasmids, transposons, molecules, morphological characteristics. In the second paragraph, we have role of microbes in soil fertility and crop production, carbon, nitrogen, phosphorus and sulfur cycles, biological nitrogen fixation, symbiotic, associative and asymbiotic, azolla, blue-green algae and mycorrhiza, rhizosphere and phyllosphere, microbes in human welfare, silage production, biofertilizers, biopesticides, biofuel production, and biodegradation. And in the last paragraph, we have important plant pathogenic organisms, different groups, fungi, bacteria, fastidious, vascular bacteria, phytoplasmas, spiroplasmas, viruses, viroids, algae, protozoa, phanerogamic parasites, and nematodes, with examples of diseases caused by them. So this course will be taught by two teachers. I will be dealing with the middle paragraph and the other teacher will take your first and third paragraph. Now to begin with the course, what is microbiology? I expect that you know what is microbiology. Microbiology is study of microbes or microorganisms. Microbes are the organisms which are very small in size, which can be observed under microscope. Now, the subject microbiology, it has many branches. Broadly, we can categorize the branches into two groups: pure microbiology and applied microbiology. Under pure microbiology, we have bacteriology, that is the study of bacteria, mycology, study of fungus, virology, study of virus, nematology, study of nematodes, phycology or algology, study of algae, protozoology, study of protozoa, microbial ecology, microbial genetics. These are the branches of pure microbiology. Then we have one important group, that is applied microbiology. There are many of them. I have listed some only. Agricultural microbiology, that is our subject here. Industrial microbiology, food microbiology, aquatic microbiology, dairy microbiology, air microbiology or aeromicrobiology, immunology, epidemiology, exomicrobiology, that is microbiology of exosphere or outer space, biotechnology, and so on. So in this course, agricultural microbiology, we will essentially study the role of microbes in agriculture. So before we go to the subject, we should know some important terms. So the terms are pathogen. Pathogen means an organism that can cause disease. Any organism that can cause disease is called as pathogen. Parasite. An organism that depends on other organism for survival. That is called parasite. Then obligate parasite. Obligate parasite is an organism that requires a living host for its survival. So an obligate parasite cannot live without a living host. Biotroph is another term to denote an obligate parasite. So obligate parasite or biotroph, these are synonyms. Saprophyte, an organism that depends on dead organic material for its survival. Obligate saprophyte, an organism that depends on dead organic material only for its survival. We also call it as necrotroph. Facultative parasite, an organism that usually grows saprophytically but can also be parasitic. Then facultative saprophyte, an organism that usually grows parasitically but can also live saprophytically. Okay. Then ectoparasite, a parasite feeding on a host from the exterior. A parasite feeding on a host externally, being external to the host. So an ectoparasite does not enter the host body. An ectoparasite remains outside the host body but feeds on the host being outside. Endoparasite, a parasite that enters a host and feeds from within. Antibiotic, I am sure you know the term antibiotic. Antibiotic means any substance produced by one microorganism that can kill other microorganisms at low concentration. So antibiotics are the products of microorganisms, and these products are used to kill other microorganisms. Penicillin, streptomycin, tetracycline, griseofulvin, these are some antibiotics. Now penicillin is obtained from fungus, but it kills bacteria. Similarly, streptomycin, tetracycline, these are obtained from bacteria, but they kill some other bacteria. Griseofulvin is obtained from a fungus, but it kills some other fungi. So antibiotics can be obtained from fungi, can be obtained from bacteria, but they do kill other microorganisms, maybe some other bacteria or other fungi. Then autotroph. Autotroph is an organism that is able to form nutritional organic substances from simple inorganic substances such as carbon dioxide. So an autotroph can fix carbon with the help of energy obtained from light source or chemical compound. So basically an autotroph is an organism which can utilize atmospheric carbon dioxide. An autotroph is an organism whose carbon source is carbon dioxide. So autotrophs can be divided into two groups again. Photoautotroph, that means energy source is light, carbon source is carbon dioxide. Chemoautotroph means energy source is chemical compound and carbon source is carbon dioxide. So photoautotroph, chemoautotroph. Then heterotroph. Heterotroph is an organism deriving its nutritional requirements from complex organic substances. They are unable to fix carbon or unable to obtain carbon from carbon dioxide. So heterotrophs cannot utilize the atmospheric carbon dioxide directly. They depend on carbon compounds, organic compounds for their carbon source. Then comes symbiosis. Symbiosis is a phenomenon of living together of two dissimilar organisms. Okay, phenomenon of living together of dissimilar organisms. Mutualism. Mutualism means living together of two dissimilar organisms in which both partners benefit each other. So there is a difference between these two terms, symbiosis and mutualism. Symbiosis means living together of dissimilar organisms. That much only. Whereas mutualism means living together of dissimilar organisms in which one partner benefits the other. Actually, both partners get benefits out of the symbiotic relationship. So in nature, we have a number of mutualistic relationship among organisms. One such example is the mutualism between fungi and roots of higher plants. And that mutualism is called as mycorrhiza. Another example of mutualism is the rhizobium and legume plants. Alright, next is parasitism. Parasitism means living together of two dissimilar organisms in which one of the two partners lives at the expense of the other. That means there are two partners in the relationship, but one partner gets the benefit and other partner is at disadvantage. Next is commensalism. Commensalism means growing together of two dissimilar organisms in which one partner is benefited and there is no disadvantage to the other partner. That is commensalism. Symbiont. Symbiont refers to the partners involved in symbiotic relationship. So when two dissimilar organisms are living together, then each partner is called as symbiont. Okay, now let us see a brief history of microbiology. How the subject microbiology began. Who started studying the subject microbiology? Alright, the first name that comes in the picture is Aristotle. Aristotle was a philosopher. He observed that animals might evolve spontaneously from the soil, plant, or unlike animals. So, animals evolve spontaneously from the soil, plant, or unlike animals. And he propounded the theory of spontaneous generation. Spontaneous generation, which states that microbes arise automatically in decomposing organic matter. So, from organic matter, microbes arise automatically. That is spontaneous generation. So, microbes or animals they arise spontaneously from the organic matter. So, that is what is called as theory of spontaneous generation. Now, this is false. We obviously know it now. No organism can arise spontaneously from organic matter. But it took long time to establish or to disprove this theory of spontaneous generation. There are many people, learned people, who supported this spontaneous generation theory, like John Needham in 18th century. He is the man who first observed the association of nematode with plant disease. Nematodes are microscopic thread-like animals or worm-like animals. They cause disease in plants. They are soil inhabiting organisms. He supported the spontaneous generation theory. There are many other supporters as well. Because of the lack of proof, because of the lack of scientific evidences, the spontaneous generation theory, it was set in the people's mind. People believed that yes, microbes arose automatically from the decomposing organic matter. Then the real breakthrough came from a Dutch cloth merchant in 17th century. He first observed that microorganisms exist. It was the first observation that microorganism exists. In fact, he discovered many microorganisms like bacteria, protozoa, algae, yeast. In fact, he discovered the microbial world with his own or self-developed simple microscopes. His hobby was to make lenses and see small objects. So, with his self-developed microscopes, simple microscopes, he discovered the presence of microorganisms, and he made drawings, very fine drawings of all the microbes he observed under microscope. So Anton van Leeuwenhoek, he is actually the discoverer of microbial world. Now the study of microbes got an impetus. Study of microbes got a boost after this discovery. Now various people from all over the world, they started studying microbes. Then Louis Pasteur from France, he disproved the spontaneous generation theory conclusively. He did some experiments and disproved the spontaneous generation theory. He gave germ theory of disease. Germ theory of disease means microbes cause disease in very simple term. So for any disease that we see, there is a germ, there is a microbe involved in it. Many other observations were also made. Microbes were responsible for fermentation. Then microbes were found to cause many diseases like anthrax, tuberculosis. Then many techniques were also developed how to kill microbes. The moment people understood the harmful effect of microbes, they started researching on how to kill microbes, those which are harmful. Then Louis Pasteur gave a pasteurization technique, a technique through which a number of harmful microbes can be killed from food items like milk, and food items can be stored for longer time. This pasteurization is still relevant for killing harmful microbes from milk. Milk is pasteurized by heating at 72 degrees Celsius for 15 seconds. Though pasteurization does not kill all the microbes from milk, but most of the microbes are killed when heated at 72 degrees Celsius for 15 seconds. Louis Pasteur also developed vaccine for anthrax. Now that was a time when Louis Pasteur developed vaccine for anthrax disease. Anthrax disease is caused by a bacterium, Bacillus anthracis. So he succeeded developing a vaccine for anthrax disease. So he did lots of experiments, lots of pioneering work in microbiology, in the study of microbes. So he is called as pioneer of modern microbiology, pioneer founder of modern microbiology. There are also many other important developments. One such development is here by Anton de Bary in 1845. He first established that fungus is a cause of plant disease. Plants also get diseased, but before him, the idea was just opposite. Microbes are the result of a disease, not the cause of a disease. But it was him who experimented with a disease called late blight disease of potato, and he conclusively established that fungus is a cause of a plant disease, not the result of a plant disease. So, his contribution also proves that spontaneous generation theory is wrong. It in fact establishes the germ theory of disease originally proposed by Louis Pasteur. So, for plant diseases as well, germ theory of disease was proved by Anton de Bary. So, Anton de Bary is called as father of plant pathology. And the fungus which causes the disease in potato is called Phytophthora infestans. Then Robert Koch, again one very landmark research work was done by him. He proved that bacteria can cause disease. Actually, he discovered the bacterium which caused anthrax disease. He also discovered the bacterium causing tuberculosis or TB. He also devised various techniques to culture bacteria in the lab. He also gave Koch's postulates to prove that a microbe is really the causal agent of a disease. How to prove a microbe to be the pathogen of a disease? So he gave some guidelines. If we follow the guidelines and the microbe fulfills all the guidelines, then the microbe will be called as the pathogen of the disease. So he gave a number of techniques related to study of bacteria. So he is called as the father of bacteriological techniques. Later on, many other discoveries were followed, like Ivanovsky worked with tobacco mosaic disease. Beijerinck also worked with tobacco mosaic disease, and he discovered virus. He is called as father of virology. Then Tort and D. Harrell, they independently discovered bacteriophage. Bacteriophage is a virus that kills bacteria. Then the real nature of virus was discovered by Borden and Piri, that viruses are basically made up of nucleic acid and protein. Then some of these discoveries are known to you, like discovery of penicillin by Alexander Fleming. Penicillin is known as wonder drug. It is an antibiotic that can kill bacteria. It was the first antibiotic discovered, and it was produced by a fungus called Penicillium notatum. It is a fungus, but the antibiotic obtained from this fungus kills bacteria. Then Waxman discovered an antibiotic called streptomycin, and this streptomycin is obtained from or produced by a soil inhabiting actinomycete or bacteria. And the name of the bacterium is Streptomyces griseus. It is present in soil, and many antibiotics are obtained from this bacterium Streptomyces. It is a rich source of many antibiotics that we use today. Then many other developments like proving DNA as the genetic material, discovery of prions. These are infectious protein molecules causing diseases in animals. Then invention of electron microscope, discovery of viroids, classification of organisms into prokaryotes, eukaryotes, introduction of the kingdom Protista, five kingdom classification of all organisms by Whitaker 1969, and those five kingdoms are Monera, Protista, fungi, Plantae and Animalia. Then invention of compound microscope, introduction of use of cotton plugs in the lab, then a technique called Tyndallization given by John Tyndall. It is a method of killing microorganisms through repeated heating and cooling of the substance. Say for example, a kg of soil or a bag of soil. If you want to kill all the microbes from the soil, then you have to follow repeated heating and cooling for a period of 3 days. So that will result in killing all microbes from the soil. Then discovery of first bacterial disease in plant, use of petri dishes to culture microorganisms in the lab, then staining technique of bacteria given by H.C. Graham, and this is called Graham staining. Then discovery of a new organism MLO or phytoplasma. These are prokaryotes, single-celled, wall-less, obligate parasites causing diseases in plants only. So these are some important developments of the subject microbiology. In fact, the history of a subject is never ending. We have lots of discoveries made even after this, and as we go along, we'll come to know many of the discoveries. So you can try to remember as much as possible from this lecture. So with this, I conclude the first lecture of the course. Thank you very much.